The third method to synthesize the radioactive probe, and the most commonly used method, is random primed radioactive labeling. As the name indicates, we use the primers which can bind randomly. अगर replication का concept थोड़ा सा ज़हन में लाइए, double-stranded DNA has to get separated. Like if you see in this picture that we have a double-stranded DNA. जब भी हमने इसको replicate करना है, then we separate the two strands, and that process is called as denaturation. And then next step is utilization of the primers which can bind to these DNA template strands, and then the strand is extended and we get the two DNA molecules from the one DNA molecule. Now the same principle is used to produce the probes by random priming method. The random priming method uses the Klinov fragment as the enzyme for polymerization. It needs the supply of nucleotides, and one of those nucleotides is going to be radio labeled. When we subject the double-stranded DNA to denaturation by increasing temperature in the presence of a mixture of random primers, those primers are going to bind with the template DNA, and they are going to extend or prime the synthesis of new DNA strand. As you can see in this picture, this is the double-stranded DNA which is denatured by heating. Now there is found only a single strand of DNA which will act as a template. In the second step, those oligonucleotides or primers which are known as hexamer, they are going to bind with the template strand at random positions, and the extension of those primers, they are going to fill up the gaps. Now this is the method which is preferred over both the previously talked about method, because of the reason this method is going to give you very high activity of the probes. इसमें रीज़न क्या है इसके पीछे? क्योंकि हम जो प्रोब सिंथेसिस कर रहे हैं उसमें कंप्लीट स्ट्रैंड जो है डीएनए का वो न्यूली सिंथेसाइज्ड है, और उसमें मैक्सिमम नंबर ऑफ रेडियो एक्टिवली लेबल्ड न्यूक्लियोटाइड इनकॉर्पोरेट हो रहे हैं. इसका हमें जो प्लस पॉइंट मिलता है हाई एक्टिविटी प्रोब्स का, दैट वी कैन आइडेंटिफाई स्मॉलेस्ट अमाउंट्स ऑफ डीएनए फाउंड ऑन द मेम्ब्रेन ड्यूरिंग सदर्न ब्लॉटिंग.